Biocurator Working Group. Today, we're going to have a presentation on NCBI Datasets by Nuala O'Leary. And we're excited about that. And we'll have some time for questions at the end. And then the Wednesday, May 8th meeting is canceled for CCG. And the next meeting is Thursday, May 23rd at noon p.m. Eastern Time, where we will get an overview of the somatic CDWG task force and VSEPs from Jason Saliba. So we're excited to hear about those updates too. So with that, I will turn it over to you, Nuala.
Okay, thank you. The screen. And can you see my slides, or are you seeing the presenter?
I'm seeing the presenter view, so just swap it.
Okay.
Looks good, yeah.
Great, thank you. Thank you so much for inviting me today. My name is Nuala O'Leary, and I am product owner of a new resource at NCBI that's called NCBI Datasets. So you may or may not have heard about it. But we've been around for about 4 years, and it's a project that's under very active development. So for today, what we will cover is an introduction to what the project is, since it's fairly new. People may not know about it, and just discuss what the goals of the project are. And it's primarily about finding and getting data at NCBI. And so I'm going to talk about, what kind of data you can get through datasets right now, and how you can go about getting that data. And I'm going to break it down into 2 different types of datasets that you can get. One is around assembled genomes, and the other is around genes, and that includes gene orthologs. And in addition, I'm also going to talk about 2 different ways of getting the data. So you can get this through our web interface, and we also have command line tools. So I'm going to go over examples of using both of those to get data. If anybody has any questions, please just stop me as I go. So data sets, as I said, this project was started about 4 years ago, and it was initially conceived to address some known problems that NCBI was having with the growth of data and the complexity of data. And as a repository, we're keenly aware of the effect that growth is having on our existing infrastructure and how that's affecting the way in which people come in and get data. So we wanted to see if we could sort of rethink the way in which our interfaces are structured, and could we create a new system that is more scalable and just easier for people to use. So our tagline here is a resource to find, download genome, and ortholog datasets for all domains of life. And just a reminder, NCBI stores data for all eukaryotes, prokaryotes, viruses. So our datasets covers the full taxonomic tree. So just to go into a little deeper, what problem are we actually solving? So for anyone that knows NCBI is a database of databases. So it has grown organically over the past 30 years. And our primary structure is that we have archival databases like Nucor, nucleotide, and genome, and protein, and things like assembly. But a lot of the databases sort of grew because new type of data came online. We wanted to structure a database specifically around that type of data. So as a result, we have a lot of different databases. And when people come in to try to get data, they're sort of traversing these databases and hoping that they can find what they need in one particular database. And as you do that traversing, you start to lose connections between data, and it becomes harder to sort of download the entire corpus of data that you need. So yes, you're probably familiar with these entree interfaces that we have. We have a programmatic tool called eutils, and that eutils works with those entree databases as a query language that allows you to extract data from the entree databases. It's very powerful, but it's very complex, and it has a pretty steep learning curve. And for people that want to get large amounts of data, FTP is generally the place that people go, but navigating through FTP is also not the best experience. So what are the challenges that people were facing? And we know that data and metadata, they're distributed across different databases, so each with its own set of rules for access and retrieval. In addition, metadata schemas in those databases are not well documented. So it's not clear even when you do use that query language, are you really getting what you need? and it lacks flexibility and scalability. For many of our databases, you can only download a certain amount of data. They can't scale beyond that. So the data sets, I just want to talk a little bit about how we went about this project. It's, it says it's 4 years old, but we knew these problems existed, but we did not want to just build something that we, conceived ourselves. We spent a lot of time at the beginning of this project talking to people, following their workflows, interviewing them. We also looked at a lot of analytics and how people were using our databases. We did usability testing along the way. So this was sort of a new approach to design. We use Agile, and we did iterative development. So we didn't want to overbuild something and release it. We did slow, small releases, tested those with users, and figured out the problems that we had along the way. So what we've come up with for data sets is a new set of interfaces that can be used to get data that is stored in the NCBI databases. So we have a web interface, which I'll do a sort of a live demo of the web interface after this section. And we also have a command line tool. And the command line tool, we spent a lot of time on this. It's a tool for getting large amounts of data or small amounts, whatever you like. But we spent a lot of time making sure that this command line tool could be used by people who are new to programmatic access. So we feel like it's very easy to understand. It's very similar to the words and phrases used on the web interface. And we feel like it's a tool that will open up this large amounts of data to more researchers who don't have really strong programmatic skills. And both of these interfaces, the CLI and the web interface, they're both powered by our API. So that means when you're in data sets, there's a single source of truth that comes from the API. It goes to the web, it goes to the command line tools. So you don't have this confusion about rules in different databases. So some of the features of data sets that we decided on based on all our testing was to create a taxonomy-focused entry to data. So we found that most people, when they were going to look for data, they used only a few terms, mostly taxonomic terms. They use accessions, they use gene names. So we wanted to have robust support for these common query terms. So if you came in with something like ACE2 or accession, you would get exactly what you would expect to get for that. We don't have a ton of filters. We have sort of common filter options, and these again were based on what people were actually doing in the existing web interfaces. So it's not overly complex, but it gets people what they need. And in the next slide, I'm going to talk about how we deliver data. We deliver data as packages. So we combine both the data, the sequence data, and the metadata together in a zip archive, so that when you download data, you have this package. You don't have to compile that data yourself. We put it all together and make it easier for better attribution of data and better connecting of metadata to data. And we also have documented schemas. So we have spent time on our documentation pages to make sure that people, when you download a metadata report, you know what those fields are and what they mean. So in addition to our interfaces, we have a new data delivery system. So I'm going to introduce you to this idea of a data package. And data packages we have ones for genomes, we have one for genes, and we have a separate one for virus because we were supporting delivery of the genomes during the pandemic. So we separated that one out. So these packages, what you can do is you have a simple query term. You can ask for the human genome. And once you get to an interface that will ask you, which type of files do you want? Do you want just the genomic FASTA? Do you want the annotation? Do you want metadata, what you want metadata about the annotation, just metadata about the genome, or do you want GFF3 type files? And then we also have metadata reports, and those reports are delivered in JSON and JSON lines format. We chose this format because it is both human readable and machine readable, and we are interested in making sure that this data can be used by data scientists, that it's scalable. So this is it's a good format for those that are building bioinformatic pipelines. We know that it's not the most user-friendly format for people to be reading, so we have a tool so you can convert these files into tabular format. So that's our data delivery system. And just to dig in a little further into what I mean by these data packages, so this is an example of a gene data package. When you download it, you'll get a zip archive called NCBI Dataset. You can open up that archive. It will have folders inside. And this example, you don't have to get everything. I'm just showing an example here where you can get different types of sequence files. So if you're requesting data for gene, you can get the transcript FASTA, the full gene FASTA, including introns and exons, protein, and then we allow you to break that down into CDS or 5' UTR, 3' UTR sequences. And then there's different metadata reports that have high-level gene information. There's detailed information on the annotation itself. And then we have a catalog that tells you it's basically a list of all the things that you got for your request. So now I'm going to switch over to the web interface. As I said, we developed a web interface that is organism focused. So this is just sort of showing a picture of what I'm about to show, but you can come in through NCBI search or come in through the datasets homepage. It will take you to a page for that species, or you can go up the tree. And from that page, it becomes sort of a central hub to data that's available at NCBI for that organism. So we know, as I said, we store data for organisms across taxonomic tree. Some have a lot of data, some only have a little data, but you can come into these pages and you can kind of see what the landscape of available data is for that organism, and then connect over to some tools. And I'll just point out this data set is a work in progress. So we have some things that we have been able to implement and distribute, and some things that we're still continually working on. So I am going to stop sharing this for a second, and then I'm going to switch over to web. And I'm going to put the URL in the chat for datasets. So if anyone wants to follow along on their own computer, do that. Okay, let me share. So let me know if you can't see anything. Okay. Can you see the page? Okay. Yeah. Okay, so this is the datasets homepage. We are, we're separate interface from the existing Entree interfaces. If you start over here, you can just come and type a name. We have auto suggest, and this takes you over to the page for the organism you searched. And this page again is the central hub for data that's at NCBI, and it's again still under construction, but the basic design is broken down into sections based on the data that's available. So the top will just have taxonomic information. There's also a section on genome. So what we highlight here is the reference genome. So that'll be the current reference genome for that species. At NCBI, we generally choose one genome per species to be the reference. There are some examples some examples where we have more than one, something like dog, where we have both Canis lupus familiaris and Canis lupus dingo. So there's some cases where we have two reference genomes per species, and generally there's one reference genome that's available. And we provide in this section a really easy way for people to just download. And again, as I described, you can open up this widget. You can choose the RefSeq copy or the GenBank copy. For those who may not know, RefSeq is the NCBI group, so it's the annotation pipeline. If we have annotated a genome, there will be a RefSeq copy of it. And then you can pick and choose the files that you want. So you're always going to get a metadata report that's included in every package. And we believe that it gets to FAIR principles, where if you're downloading data, you always should be able to cite that data and have the proper information on what you're getting. So, and then you can pick and choose the files that you need. We also have an easy way for you to go to some tools like our Genome Data Viewer. I'm not going to go into any details about this today, but if you want to hear from the team at NCBI that does that, I can connect you. So that's just a graphical view of the annotated genome. There's also a connection to BLAST. And this is a new table that we created, which allows you to view annotated genes. So it brings you over to a table of all the genes that are annotated, all the genes that were annotated in that last annotation run. So as clinical curators, you probably know that NCBI annotates genomes. And once we've done an annotation run, that's sort of a snapshot of the annotation. And that's what you're going to get in this table. It's going to be a list of genomic coordinates, their location on the chromosome, their gene type, and you could connect over to transcripts and proteins. We have just a few things you can select. If local tag are important, you could add those. And you can download the table, or you can download the package again for, you see an option here to go download the table. You can get one sequence per gene or all the sequences, and you can download a package. Again, you get to choose the FASTA files, and you'll get that metadata file. We also have an option to filter. So you could filter by gene symbol, and it will bring, it searches gene symbols and gene names, so it'll bring back anything that will match that. You can also search by genomic location. So you could take just this genomic location, put it here, and you could increase it. So, and it will give you all the genes that are annotated within that range. And if anything comes over, we'll bring you that whole gene. So this gives you the annotation snapshot, again, of the latest annotation. But we also have a curation team. So they are constantly looking at the annotation, the products of the annotation pipeline, making updates, making changes, adding additional transcripts and proteins. So when you go down to this section of the page, you get to a section called gene, and that will be that current gene set. So it'll include genes that either didn't make it on the last annotation run, or genes that were recently updated. So if you go to this table, it looks similar, but it's got richer information that is information that the gene database pulls in, and we pull from them. So when you look at this view, you have more columns that you can select. So you can select genomic coordinates, you can select OMIDs, nomenclature authority. There's different things you can select, and you can add here. And the coordinates we have here will be the coordinates that we have for that gene annotated on both the reference assembly, and then we've annotated the T to T assembly, which I'm sure you know about. So we have the coordinates on that. We don't have coordinates on 37 here, but I'd be curious to know if that's something that people would want to see. So you have options to select columns, and then again, you can select here, you can download just the table, or you can download a package. And again, this is a huge package because it's the entire all human genes, but you could select sequences, transcripts, and proteins. And just one point, the reason this number is so incredibly high is because of we're bringing in all gene IDs, so that includes gene IDs for the functional elements, the non-genic regulatory regions that we curate here at NCBI. So those will also be included in this table. So, and the last section of this page has these database links. So as datasets continues to grow and we provide more data, more packages of data that are available, we'll continue to develop this page. But right now, we're just connecting over to databases for those things that we don't yet deliver through datasets. At least you can get over to the Entree databases as we sort of continue to develop. And for the clinical resources, we link here to dbSNP and to ClinVar. So that is our sort of our taxonomy entry point. If you wanted to go higher up the tree and you wanted to find organisms, say, related to human, we have it's breadcrumbs here, and they allow you to just sort of select. You can go up the tree, you can go up to primates, say, and you'll get a page for primates, again, taxonomic information. The genome section will just link out to all the genomes that are available. So here, you can select here, go over, and what you'll end up with is a genome table. So you'll have a table of all the primate genomes that are available. We have, again, basic filters. So you can just filter down to the reference genomes here, and you can add additional columns. These are columns related to these assembled genomes. Some of them are going to be annotated, some of them will not be annotated. If it's a reference genome, it's not required that it be annotated. So we have a column here that tells you whether or not, if you go for that genome, whether or not there will be annotation files available. And then you have options to link out to BLAST, our genome data viewer, and to view details. So if you wanted to know more about the individual genome, you can select here. And this page will take you over to all the aggregated data around that individual genome assembly. So these pages are, we've been spending a lot of time on work on these pages and making them, making them better aggregates of data than the current assembly database. And in June, if people don't know already, we are going to replace the assembly resource with NCBI Datasets genome resource. So the assembly resource, the web interfaces will no longer be available at the end of June. So this page has information on the assembly. Again, links out to the annotated genes and the tools that we have. It provides statistics, and we now provide statistics for the RefSeq and the GenBank. These, in this case, are very similar, but sometimes they're different. We have annotation details, quality analyses, a chromosome. If they're chromosome level, link out to these, and these will link out to the viewer. You can download a table of the individual components of the assembly. And lastly, there's a revision history table. So if you are working on a previous version of a reference assembly, say you were on the last version of 37, you can go down to the revision table. You can either click over to it and get to a similar page, or you could just, again, download the data here if that's what you needed. So we keep that, that is the path into the previous assemblies. Or you could just search by the name of the assembly or the accession. So that's the assembly page, and we are working on improving our publication linkage. Right now, these are coming from the assembly database, but we're working on making that a little bit better. And if you want to go back to the organism landing page, just click on the organism name, and you will be back over here, that assembly, at that taxonomy page. So, and just again, to go up the tree, genomes. You can view in this table, but you can also just select one of these organisms if you were interested. You can come over here and see, again, the reference genome for that organism. You can click over to the current gene set, includes genes that are currently new or not annotated. And again, the database links for any organism. That's all. That's NCBI has data for. So if you are interested in just coming in, not by taxonomy, but you want to come in by a gene name, something we do have this menu here. So you can come over to gene. It has a very good memory. It'll remember what you searched. And here you can search either by symbol, and that will get you over to that gene table that we talked about. You can also just search by organism name. Again, and you'll get to a table that is all the genes that we have for that organism. You can also search by an identifier, or you can upload a list of files. So it'd be a text file of gene symbols or gene IDs or identifiers. The identifiers that we accept are gene IDs and RefSeq, the NMRMP accessions are also accepted, and that will bring you back to a gene record. And this gene table will also allow you to connect over to the gene resource, which I'm sure you're familiar with. Right now, this is the existing gene resource, which has a lot of information, and Datasets is working with there's an initiative at NCBI called the Comparative Genome Resource, and we are looking at redesigning this gene page to make it more modern looking and highlight additional data like expression data, Go terms, and looking at a way for making this page just a little bit more useful for users. But right now, if you're on this page, you can download from Datasets. So it's the same thing I showed you on the table. You can just go through that button. And you can also connect over to orthologs. So I want to go back to this table here and point out one more column here. So we have this ortholog set column. And what this is a set of related genes that are calculated by NCBI's genome annotation pipeline. So that pipeline takes eukaryotic organisms that are annotated by RefSeq, and it looks for orthologs based on reciprocal protein blast, and it also takes in synteny into account. So if you select on that button, open here, sorry, you will get to this page. So this is a page for orthologs for any particular gene. And what it does is give you a tree and a table here that shows you all the genes that have been placed in that ortholog set. And when available, we will show you the protein architecture for that gene. This is calculated by another group within NCBI, and we will show you. So you can click on there, and you can see the different domains of that particular protein. And if you open this up, you know there's many, many variants. If that architecture is available, it will be here, and you can click and select it. And what you can do from this page is search. So you can search for a taxonomic name, and you can narrow down your table. You can also select a few. You can select 1 or 2 organisms or more, and then you can do a protein alignment. So it takes you over to Cobalt, and you can do an alignment from here. And that will allow you to, take those different orthologs and do a sequence comparison and sort of dig in further. So you can dig down into the sequence. So the ortholog set that we calculate is pretty rigid. And if it can't just pick between 2 genes, it will just drop it from the ortholog set. So what we did to be able to expand the connections between these related genes is we created an additional web interface called similar genes, and you get to it over here. And what that does is take you to a page where genes are connected by their protein architecture. So if a gene in the RefSeq set shares this protein architecture, it will be pulled into the set. So it expands the number of genes. It goes deeper into the tree. This goes beyond just vertebrates, which is what the ortholog set is restricted to. And it will allow you to browse through these and see related genes in other species. And it brings together the data that you would find in CDD, a protein database, and kind of gives you this central view of related genes based on either orthology or by protein architecture. Let me go back over to datasets page. So the last thing I want to talk about in terms of the web, and then I will go back over to my PowerPoint to talk about the command line tool, is when you're on this page, I just want to point out one thing up here. There we have a documentation page. This is a place to come to if you want to learn more about how datasets works, basically like how if you want to do programmatic access, this is the place to come over and to understand what tools we have available, how to use them, some very quick guides. So I'll show this in the PowerPoint, but I just wanted to make sure people knew where this was, because what we find in our user interviews is that even people that want programmatic access, they use the web to sort of understand the data, see what's available, and sort of explore before they go and use the command line tool. So one of the things that we've done to help people with that transition is if you are on this page for assembly, there's these 2 things at the top here. This is datasets, that's our command line tool. So if you click on it, it actually gives you the command. So you can take that over to your terminal, and you can get started right away. So once you've done the exploration, if your needs are really to download a lot of data or to work with that data as a data scientist, this is the way. You should go over to the command line tool. It's the best way to get the data. So I'm going to stop sharing once more, and I am going to go back to my PowerPoint. Okay, I believe that's right. All right, so now I'm going to talk about our command line tool. So we have 2 command line tools. The first one is called datasets, and that's for querying and downloading sequence and metadata. And then we have a second one called data format. And data format is for taking that JSON or JSON lines files that we have and converting them into TSV or Excel. And so, as I said, we've tried to make this command line tool very easy to understand. So this diagram here shows the basic structure of the tool. The tool's name is Datasets, and you have two paths you can go down. You can download data, or you can do summary command. And the summary command is just going to print metadata to the screen. And much like our web interface, it's broken down into genome, gene, virus, and taxonomy. So if you go down each of these paths, you're going to get data related to the assembled genome, to a single gene, again to the virus, or to just taxonomy. And it reads sort of like a sentence. Once you go in, you have to pick how you're querying. So datasets, genome, accession, you can query by taxon. And then we have certain flags and filters that you can use. But it's a long command, but it's easy to understand. It's kind of the same across the different services. And it takes in the terms that we largely see people using. Down here, rehydrate is, I won't talk about this today, but this is a path that we created for people that are, if you're downloading, say, hundreds or thousands of genomes, we recommend going down this path that allows you to download the metadata plus these fetch files, and then you run the rehydrate command, and it downloads in parallel. So it's a much better experience if you are downloading very large amounts of data. So that's the tool. So I'm going to go and talk a little bit more about using this tool to get gene data. So again, you can enter when you enter this tool, you go through your path, and you pick whether you want to download the zip archive package, or you just want to look at the summary data. Again, you pick your symbols. It's the same that work in the web interface. And flags that we have are, we have two different types of reports. You can get just high-level gene report, or you can get annotation reports. So the report string is to get those different types of metadata. Taxon string is if you're using gene symbol, you have to tell us which, by default, you'll get human, but if you want to download by gene symbol, you have to tell us what organism you want to download for. And then we have an ortholog string. So you can put in human BRCA1 and then use the ortholog string, add a taxon term after that, and get all the orthologs for that. So I'm going to walk through one command. This one is going to go datasets, download, gene, by symbol. And then again, here's some flags. Once you've entered the symbol, you can pick and choose, again, those files that you want, and the different flags. So here, this is the download. So I've just put in the symbol, and I'm downloading by symbol, and I put in a list of things, and I name my file. I unzip it. What you're seeing here is just a default report. You're going to get the RNA files, the protein files, that basic data report, and then a catalog, which will have a list of all the gene IDs, basically a list of everything that's in your package. And so that's the download command. If you want to use the summary command, it's again summary, gene symbols, same structure. And I'm piping it to JQ. JQ is a tool for working with JSON lines. So we wanted this, this output that we have to be interoperable. So these are tools that are out there that anybody can use, and they will pretty print the JSON output into this format. So this is what JSON looks like, and so it's easy to read, but it's not tabular, so it's not the most user-friendly way of looking at data. So to make it more user-friendly, to generate into tables, we have a tool called data format. And data format is a tool that you can use with the summary command. You're going to pipe it to data format, and you can use this fields command. And the fields will take those data reports, and on the web, you can see our data reports. They have all of the fields that we have in our data reports. Pick and choose fields that you want, or you can pretty much take everything. And so you have gene ID, gene type, fields, and that will, I'm showing the example here where I picked a couple of fields, and you will print them out here. So you use that, and you need the summary command prints out in JSON. We have to convert it to JSON lines, pipe it to data format, and then the fields. And so you can generate a tabular report of anything that is in that metadata report. So we think that's an easier way of people working with these JSON lines. If you're on the web, you can obviously download the table, so you don't need to go through this. But for the command line tool, this is a way of converting it. So For our command line tool, as I mentioned, we have documentation pages. We have how-to guides. These are really short commands that you can just use to get started, and they should work, and you can see the results and start playing around with it. We also have these longer tutorials, and these are things that we've used in workshops and other places. So these are more like guiding you through different steps to carry out a particular task. And this reference section here, this is where we have a description of all the files, of all our metadata schema. You'll also find our API docs in that section there, if you are, strong programmatic skills and you want to use the API. So lastly, we just want to point out that Datasets is also in Galaxy. We've been working with Galaxy over the last couple of years to integrate our tools into their interface. So you can, I'm highlighting the gene service here. There's also Datasets genomes. So if that Galaxy is an environment in which you like to work, you can go ahead and use it through there. And just to end, what's next for Datasets? So as I mentioned, this is a project that is under very active development. So we are constantly getting feedback and working on improving our existing services and then expanding. So some of the things that we're looking at that I mentioned already is that we're looking at redesigning new gene pages. We're expanding the ortholog set to go into more to invertebrates. And we're looking at incorporating Go data and expression data on these new gene pages and through our downloads. So that's something that we're exploring now. And we're also looking at our improvements to our metadata schemas. This we've gotten a lot of feedback on, and we want to make it as easy as possible for people to get that data and also to filter through it. So that's what we're working on. And last, I just want to thank my team over here. This is the Datasets team. And over here are people within NCBI that have contributed to this resource. So with that, I will stop and take any questions. Thank you all very much.
Thank you, Nuala. That was great. So I think we had one question in the chat. Can users get alerts for specific genes if they've been re-annotated?
Alerts for specific. We don't specifically have that in Datasets. So that's a really great. So when a new gene has been curated or additional transcripts added, you want some alert that there's some sort of update. That's we don't have right now, but that's something we could maybe incorporate into the new page. We were thinking with the new pages that we would have the annotation snapshot as sort of the primary thing that you're going to see, and then an updated section. And maybe when we develop that, we could also have that be some sort of alert process. So, thank you. Great suggestion.
And then Deb, do you want to unmute and ask your question?
Sure. No, I was just wondering for the command line that you were showing, that isn't something that you can do by putting it into a browser, correct? You need to download a utility of some kind or use Mac terminal and do that.
You do, yes. So
yeah, okay.
Share it is we have it is available in Conda. A lot of people like to use that environment. So if you go to our command line tool page here, it gives you all the options for installing it.
Great.
Download it, yeah, and you can install using Conda. And just to point out, we also have a GitHub page here, and on the GitHub page, we've run longer tutorials and trainings using Jupyter notebooks. So the last one we did was at the Tag C workshop. So if you come to our training page, what you can do is this is sort of you can read through this tutorial. I didn't want to present it here. It's sort of long and hard. It guides you through many aspects of the command line tool and follows sort of a story, sort of similar things. And
that's wonderful. Great. Yeah. Thank you.
You're welcome.
Could you put that link actually in the chat? It would be fantastic.
Sure.
Thank you.
Yeah.
Great. Thanks, Deb. And then we have another question while you're sticking that in the chat. How are annotations and nomenclature handled for genes with multiple functional copies?
Genes of multiple functional copies. So, like gene duplications, is that what I understand?
Dimitri, is that what you mean? Yeah. Yeah.
So for human, I mean, for nomenclature, we take nomenclature from HGNC. That's that's the authority that we follow. In general, if there is a nomenclature authority, NCBI will follow that authority. And so what we've done in data sets, at least, is try to provide in our tabular views and in our reports as many identifiers that come from the different authorities. So we have ensemble identifiers, HGNC, and any other to make sure that those gene IDs can be connected to those. Not exactly your question, but I think we follow HGNC, and so it is flexible in that something has like a core name. You can search by that core name. If there's 1, 2, 3, 4, then you can get a tabular view of that core symbol name.
Great, thanks. Does anyone else have any questions? Either feel free to unmute yourself or stick something in the chat. Okay, if not, Neil, I want to thank you for your time, and I want to thank everyone else for attending this call. And we will see you all in a month's time to hear about the somatic efforts. And I hope to see some of you or many of you at Curating the Clinical Genome, either virtually or in person in Baltimore in May. Thanks, everyone.
Thank you all very much.
Bye all. Thank you.